So morning, everyone. Welcome to the next installment of the JKMRC Friday morning seminar series that take place here at the Indooroopilly Lecture Theatre and online. And of course, today is the first installment of the seminars for semester 2, the current semester. So my name is Katerina, and myself and Karina are co-chairs and organizing the seminar series and are taking turns introducing the speakers this year. So before we start, on behalf of the University of Queensland and the Sustainable Minerals Institute, we would like to respectfully acknowledge all traditional owners and their custodianship of the lands on which we all meet today. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australia and global society. So our speaker today is Dr. Jennifer Cooling from the School of Earth and Environmental Sciences here at UQ, where she's currently working as part of the palynological/deep time research group. She completed her PhD on the palynology of the Jurassic-Cretaceous transition in the Surat Basin. And since then, Jennifer has been lured up into relatively more recent paleoenvironmental work on the tertiary, in quotation marks, sediments of Queensland. And now she's also working with the last few thousand years of history of the southwest of Tasmania. So for all of those who don't know what I'm talking about, this is geological humor, okay? Today's presentation is entertainingly titled Between Dinosaur Salads and the Ice Ages and the Difficulties of Studying the Paleogene and Neogene in Queensland. So please ask lots of questions here in person and online, because we all want to understand what it all means, okay? Thank you. Take it away, Jen.
Thank you. So hi, as Carrie said, I'm Jen, one of the postdocs currently at SEAS, doing general dogs body stuff for Patrick mostly, but helping out with the students, working on that Tasmanian core. I'm trying to get together things to put together, a proper grant proposal to work on what I'm going to be talking about today. Because as mentioned, I did my PhD on the palynology of the Jurassic to Cretaceous transition in the Surat Basin. This was a project chosen to basically train me up for succession planning with my co-supervisor, John McKellar, in how to do biostrat and stratigraphy in Queensland. Basically what I did was, if you see, we have the chronostratigraphic, biostratigraphic, and lithostratigraphic units. I slightly wiggled a few of those lines around. It turns out that original plan of what I was going to be doing next kind of fell through. And I also found the whole, as well as just using your palynomorphs as useful ways to correlate and date rocks, working out what they actually came from and what that told us about the environments of this time was pretty interesting. So subsequent to that, I'm now working with Patrick at fairly modernish materials, but in between, during the events of 2020 and 2021, I wound up working with mostly Joan Essley here and Tianjiao Yu, one of our then honours students, now a PhD at QUT, on a core taken through a Cenozoic channel that's cut into the northern part of the Bowen Basin. So here's me and Tianjiao with some of the core that BHP drilled. We initially logged it, we went out, did some more logging and sampling, and Joan and Tianjiao did most of the heavy lifting in terms of understanding the geology, the clay mineralogy, which is a strange and mysterious topic that I'm really glad someone else was happy to do. And I was just meant to look at the palynology, go, how old are these sediments? And maybe do a little bit of waffle about what the environmental implications of that was. So here is the core log put together by Tianjiao. As you can see, it got litho logged and a lot of, what's the scanning topic thing again? I've just, hyperspectral happened. As you can see, I acknowledge the importance of other people's specializations because, all right, but basically we have this core. It's just under 100 meters, and at the base of it, we intersect that, classic Permian coal majors. Just above the base, though, you have a unit of breccia that rounds up into a conglomerate before you enter this sequence. So that was unit 4. Unit 3 was an alternating series of lighter and darker, very fine-grained rocks of generally more or less organic preservation. On top of that, we have the unit 2 sequence, which is lithologically a lot more mixed, but you have more sands, more evidence of paleosols and that sort of thing before you have a couple of basalt flows come over the top and just sort of seal all this in. So the question was, palynologically, how old was it? What sort of stuff was in there? We also initially had the question of whether or not all that in the middle was actually sedimentary sediments, or if it might have actually consisted of a lot of heavily weathered igneous material. And first, the fun palynology results. This is from unit 3 and 2 and shows the relative proportion of a lot of the key taxa. So in terms of what this told us, it did tell us that there does seem to be a general change in the environment between unit 3 and 2. Unit 3 has, in general, more species associated with rainforest and type environments, and it has a greater diversity of those species. On the far left, you have various forms of Nothofagus, like the most characteristic rainforest tree of the Paleogene and Neogene. And you can see, whilst your absolute number doesn't necessarily change, you have a real diminishment of the diversity in the Nothofagus flora as you move up into unit 2. You also have what is arguably on a whole an increase in the abundance of pollen from plant groups like the Proteaceae, Casuarina, and Metaceae, so those that are generally considered part of the drier adapted sclerophyllous vegetation today. And we think this, combined with the work from the lithological logging and your clay mineralogy, indicates we're catching some sort of drying trend here. So the other big thing we wanted to know from the palynology was, how old was it? And this was the less fun results. So unit 4, I determined to be definitely Cenozoic in age. But honestly, that one I was pretty happy with, just being able to go, Cenozoic rather than Mesozoic or pre-Mesozoic, because we had to get that by scraping out bits of the matrix between the clasts and trying to get a few syn-depositional pollen grains rather than just the Permian again. Unfortunately for my confidence as a budding biostratigrapher, for unit 3 and 2, I determined it was somewhere between Eocene and Miocene in age, which, if you haven't looked at the geological timescale recently, is kind of a big chunk. Like, that's quite a lot of time. And coming from that PhD project where it was getting a lot closer to an actual, useful number, this was a bit of a blow. So what was my problem? Well, one, working on this during the time meant it was harder to find good copies of the literature that describes the species, et cetera, and angiosperms are weird guys. They hadn't shown up yet in the Jurassic-Cretaceous, but, that wasn't really the big issue. But it wasn't just me. It turns out Terrestrial sediments in Queensland and the rest of Northern Australia are in general very difficult for anyone working on them to put an accurate and reliable date to. There is a fantastic biostratigraphic scheme for Australia in this period. That doesn't really seem to work up here. And absolute age dating is expensive and also generally requires fairly specific minerals to be present in your samples to be able to do it. So quite a few minutes in, here is sort of the belated key themes of this presentation. Terrestrial sediment dating in Queensland, hard. It would be good to be able to do it because this period in between when the dinosaurs catch an asteroid to the face, the end of the Cretaceous, and when you have the real ice ages start, the Pleistocene and the Pliocene. This is sort of when Mesozoic Gondwana turns into the Australia we see out the windows today. But I think there are a number of promising leads that could pave the way to actually being able to significantly improve our ability to do age control in this period. So a quick geochronological refresher for anyone who hasn't looked at this time period lately and is going, wait, what's the difference between the Paleogene and the Paleocene? So in general, everything, if Triassic, Cretaceous is the Mesozoic, everything since then falls into what we call the Cenozoic. Next level down from that, you have your, skipping Tertiary for now, Paleogene, Neogene, and Quaternary. So Paleogene is the 66 to 23 million year ago age. So post-dinosaurs, very much a still greenhouse type world. Down the bottom here, we have the benefic oxygen isotope curve, which shows broadly, globally, temperatures compared to today. So coming out of the Mesozoic into the Paleogene, you do have what is effectively described as a greenhouse world. There's no evidence of major ice anywhere. The poles are ice-free. You appear to have had actual forest ecosystems growing at those high polar latitudes where they're having to experience periods of permanent sunshine for months and then months of permanent darkness. I don't know, that's just a completely alien type of vegetation to anything we have today. But then as you pass from the Paleogene into the Neogene, you can see that the ice sheets are starting to build up, and we're starting to move into what is a more modern type of climate regime where you start, they haven't really fully kicked in the Miocene, but by the Plio and Pleistocene, you're really starting to hit that glacial interglacial cycle and what we would consider the modern climate parameters have established. So climatically, some big important things happen. People consider parts of this fairly interesting from the point of view of what has climate change in the past looked like, in particular around the Paleocene, Eocene, where you have the most recent thermal maxima. So it's a cool time. The term I'm probably going to use a lot rather than just saying the Paleogene and Neogene to refer to the bit in the middle here is Tertiary. It's an old geochronological term, but it is kind of useful for going this bit. All right. So why did I think initially putting a nice age date on these samples was going to be easy? Was because I was fully aware that in Australia, a lot of big biostratigraphic work has been done, and that we have a great biostratigraphic zonation that extends down past the Permian and up through into the Pliocene and Pleistocene. In the time period we're looking at, this is the sort of latest version kept on GA's website. it's based a lot on work of Stover, Evans, Partridge, McPhail, and based largely on the work in the Gippsland Basin. So if on, you can see you've got for the spore pollen zonations, an ability to generally get a reasonably precise age according to this, by biostrat terms at least. And there's a lot of key events of when species are appearing, disappearing, reaching acmes of abundance, etc. And this is all based on work in the Gippsland Basin, otherwise known as God's gift to Southeast Australian palynologists. It is an incredibly prolific hydrocarbon province. It has undergone a massive amount of exploration over the last more than 50 years. I think last time I checked on GA's website, there's been over 300 exploration wells drilled, tens of thousands of kilometers of seismic, and as largely a mix of terrestrial and nearshore marine sediments, the best way for people looking for hydrocarbon resources to work out if they've drilled into a prospective area was biostrat. And therefore, a lot of money got put into doing a lot of biostrat. And the scheme is great. Like, I really wish it worked. And on a national level, it really has helped us develop an understanding of how the Australian vegetation goes from that Mesozoic Gondwanan type rainforest through a number of major vegetation changes towards what we see in the present. The most recent big review on all of this is History of the Australian Vegetation, edited by Hill, published mid-90s, slightly updated in the early 2000s. And this is genuinely a great book, and I thought it meant my work was going to be reasonably easy to date. However, if we look at a couple of figures from that book, you'll start seeing where the problems are. These are showing where a lot of your samples have come from, and you might notice some parts of the continent are much more filled in than others. Like there is a certain directional bias to our record. If we look at this one, which shows a lot of the Neogene pollen sites as well, you can really see that Southeast Australia, we have a lot of data, because once all that industry work happened and got that good biostratigraphic scheme, people working in the area could then apply it to the random bits of rock they found, for more academic purposes. So the scheme works really well in Southern Australia. Unfortunately, the tertiary basins of sediment of Queensland, we don't have an equivalent of the Gippsland, where you have a near continuous record from the late Cretaceous through to nearly now. They have been generally viewed of being of fairly minor economic interest, therefore not that much study has gone on relative to some areas, and possibly the bias is also down to it's less convenient for researchers often based in southern universities to come up and get samples. The other problem, people have been working on this section, and it turns out, all right, biostratigraphy is great. I'm not just saying this because I do it and I would like people to keep paying me to do so, but it is honestly one of the best ways to date a lot of clastic sedimentary rocks that are terrestrial in nature. Unfortunately, to do biostratigraphy, you need plants to make your index fossils. And when you're working in reasonably similar areas, you can generally assume plants are appearing and disappearing from the geological record at, by geological terms, more or less the same time and in the same order. Unfortunately, they are plants, and therefore you can only stretch the schemes so far. So Australia from top to bottom is a bit over 3,500 kilometres. If we look at the climate zones across the country today, there's quite a few. And as a result, these latitudinal differences, the plants that grow in different, top and bottom half of the country are fairly different. And on a geological term, they arrive and disappear at different times in different orders. And some of the key biostratigraphic indexes from the Gippsland Basin just never show up or are known to show up in weird orders, which is why it's always been our understanding is Southeast Australia's paleoflora, pretty good. Everywhere else, question mark, question mark, question mark. Quickly addressing the issue of, does it have to be biostrat? Not always, and absolute age dating is really good, particularly whenever you can do it. Fortunately for the tertiary period, the most convenient sediment dating methods being radiocarbon and luminescence type techniques don't stretch back this far. And a lot of your other geochronological techniques require fairly specific minerals to work. Uranium lead is great if you have a ashfall that's landed in your sediments and dropped some nice fresh zircons in. You can date those. Here's one from the paper Joan Tianzhao and I've worked on that I thought might give us a really good, independent age control. It got reworked. It's from the Permian. But it looks so pretty. I was sure this was going to be 20 million. Nah. So if your sediments are overlain, underlain, ideally interbedded with igneous stuff, that's great. But a lot of sediments aren't, and therefore we do kind of have to rely on the biostrat. Just a quick aside to that third point of my points for this presentation. There is a lot of volcanism that happened in Queensland during the Cenozoic, a massive amount, which means there are a lot of places where people have described, although they haven't necessarily done bio, pali, and geochronological dating, that you have interbedded igneous and sedimentary units. I think if we start getting a good framework for a biostrat scheme, this is going to make it fairly achievable to tie that in to actual geochronological ages. So that is a source of optimism for me. Vasconcelos et al. Have done a big review on all the different geochronological methods available for the period. It is a good paper if you're interested in that sort of thing. All right. And I do want to point out that while I've said doing palynology type stuff in Queensland is hard for the tertiary, this doesn't mean people haven't been doing it. This is a incomplete list of some of the bits and pieces people have done. So we have a lot of the pieces, of the puzzle of what vegetation change in Queensland could have looked like if we could just start actually lining these up together. So this is all stuff people have done. I'm not going to quite say for fun, but without necessarily major economic drivers for a lot of it, but going. We have a lot of the pieces in a pile, but we just need to be able to actually start lining them up to make a cohesive story. So coming out of my finding that the tertiary is hard, and that this is in fact a problem people have been talking about for the last couple of decades, there is that surprising amount of tertiary sediments around Queensland. This is from Geology of Queensland and shows a bunch of the known semi-mapped tertiary basins. A lot of these have received fairly minimal study, but we do know there's a fair bit of sediment in there. There is material you could pull together probably a biostrat 4. We might not have that equivalent of the Gippsland where you have like a couple of key cores that'll take you all the way through. But I think in all of this, you could probably string things together and get a good biostratigraphic zonation. Like, it's an achievable target if the work gets done. And my suggested launching point would be starting in the Jurassic Basin. So the Jurassic Basin is one of the oil shale basins in central coastal Queensland. Age control on it is a bit mixed, but it's clearly of Cenozoic age. And there are a number of GSQ stratigraphic boreholes that intersect it. 2 of them in particular intersect each over 1,000 meters of Cenozoic sediments. From the report, palynological study was planned for these, but as far as I can tell, it never actually happened. It was an awkward time in Queensland Biostrat. So like these cores are still sitting out at the Exploration Data Centre. Like, that's 1,000 meters of sediment. That's probably going to cover a good chunk of this time period. And from there, you should be able to start going. This lines up with this one and start piecing things together. So I think at this point, we do have the key things we need to actually create a good biostratigraphic zonation adapted for the Queensland circumstance. It just needs to get done, as in work applied. Like, we have the raw materials to work from at this point. So for the last bit of the presentation, I just want to go through why the tertiary matters. So these 2 images are really nice. They're from Peter Trussler and Monash, and they sort of show before and after this period. Beforehand, you have the Mesozoic with your dinosaurs. Afterwards, you have the Pleistocene, bit of the Miocene, with your, that really charismatic megafauna type stuff. And this is sort of the periods that are, in terms of paleontology in Queensland, the cool stuff. This is what people, when they're 5, go to the museum to learn about. But, just looking at these 2 images, you can see that there's quite a difference in not just the animals, but the habitats they're occurring in. Like, this is that time period in which we go from the last bits of Gondwana as a whole to Australia today. So some like tectonic highlights through this period. Australia and Antarctica finally finish breaking up. Australia starts getting closer with its northern neighbours. Coral and Tasman seas open or continue to open. Lots of volcanism. Possible movement on the Great Escarpment. I am not good enough at tectonics to give an opinion on when exactly the Great Divide was a thing, other than it appears the answer is complicated. But, geologically, there was a lot happening. And biostratigraphy can help us better understand that sort of thing. As a whole, the continent, basically Australia moved an Australia length up in this period. So, we start down pretty close to the polar latitudes and end up where we are now, which I think has an interesting impact on Australia's response to the global climatic conditions that change. So overall, the big trend of global climates through this period is everything gets cold. You have ice ages start coming in. But in Australia, that's slightly offset by the fact, as it's getting colder, Australia keeps scooching up into warmer and warmer latitudes. So we maybe had some periods in which you had glaciers forming in maybe the highlands of Tasmania, but we never really had an ice age. We didn't have those sheets of ice covering the continent. Probably the biggest impact of these globally cooling climates is Australia starts drying out. We think Australia as a whole sort of dries out from the middle outwards. But again, because we have that very biased record of plant history, it's pretty hard to quantify when and where this drying trend was occurring. So with that section I showed of our possible drying trend, there were a lot of periods that could come from. That's not necessarily the big signal of the final rainforest retreat from that area. There were probably phases, but a lot of our ability to date this stuff is very much being, we see this trend, we are therefore assuming it is happening at the same time as it is happening everywhere else. And you get into very circular reasonings for when you're trying to put these pieces together. So vegetation-wise, you see this big shift from widespread mesic communities adapted to lots of rainfall fairly consistently through the year to either drier or more seasonal climates. The big terms that the paleobotany people like are the sclerophyllous and xeromorphic adaptations. There's evidence that the foundations for these were sort of started being laid down in as early as the Cretaceous. They may have been initial adaptations towards growing on less good soils effectively that meant some plants were more predisposed to being able to survive under those drying conditions once they started arriving. Also interesting for people that you could be getting from a better paleo understanding is when certain plant families moved into Australia or spread out from Australia. We know probably a lot of the contact was coming from the north, but we're relying on picking up the evidence on showing up all the way down in the south. And the other big change that could actually have driven a increase in your actual megafauna was you were probably seeing a shift from generally more closed vegetation across a lot of the continent to increasingly open vegetation. So I'll just quickly run through what we know of some of the big vegetation highlights based on that work in Hill. So Paleocene, one of the coolest periods of the Australian Cenozoic. We don't have any really good KT boundary type sections in Australia, but we do know that once we start getting evidence again, you actually have a period of more conifer-dominated rainforests compared to the more angiosperm-dominated stuff we had at the end Cretaceous. Possibly it appears that angiosperms may have been hit harder by whatever happened in Australia around the time of the KT, and some of your older plant groups were better predisposed to being able to be those first expanses into disrupted habitats. Coming up into your Eocene, so that warmest and wettest period, you have evidence that we had up to megathermal rainforest extending down to Tasmania. This is just such a different environment to what we see. But again, you're probably looking at a fairly heavily forested Australian continent as a whole, although a few more points of evidence would make it a lot easier to see this. As you start moving up into the later part of the Paleogene, you start having those nothofagus rainforests, which are so characteristic of this time period, with a lot of your plant families that are going to turn out to be really important to the modern Australian vegetation starting to become more dominant components of the vegetation and starting to really diversify. And, this is where you start seeing, at least in the southeast, more evidence of open habitat type situations. As your cooling trend continues, we think, based on the few closer to the middle records we have, you're starting to actually see the impacts of things starting to dry out and a switch to more seasonally adapted climates. And then, you go through a bunch of complicated periods where you have lots of changes and stuff starting to swing back and forth as the climate doesn't have just sort of one big tipping point into middle's a desert, the outside's a bit wet. Like, quantifying this would be really good. And this is all what they've managed to do with a really biased record. Think what we could do if we had more of those pieces we could put in and go, actually, whilst we're seeing this trend here, up in Queensland, we're finding, no, there wasn't really any big changes happening at this point, whilst down south, you might have wildly changing vegetation types. Yeah. So I think we are running out of time, so I won't make you sit through all the how plants change. But basically, this is such an interesting time in terms of turning Australia into the continent we see now. And it has been fairly underserved in terms of research. And I think we could probably change that. Anyway, that's all I had by way of presentation. So do I just go question?
Thanks, Jen, for this. I truly appreciated the power of geological timescale, but I think I forgot. Do we have any questions? We do.
Yeah. What was the time period that the megafauna all check out? And is that captured in these core?
Stole my question.
In this particular time period, in the Paleogene/Neogene, we don't have the disappearance of the megafauna much. What we do interestingly have is where the megafauna came from. So like at the end Cretaceous, mammals were really small, tiny, kind of insignificant things. We get to this, like you have some wild and wacky things happening. But we're not really sure how you go from it's a rodent to all right, it's a really big mammal, but like this is that period where like the megafauna extinction is really interesting, and I understand it's a key debate. But also interesting is, so how do we get megafauna? Like what conditions drive the appearance of these really weird and wacky animals? And, how did the megafauna live while they were actually the main part of the ecosystem? So we know about the megafauna dying, not as well as we should, because age control through there is also not historically great. But the actual environments in which they arose and existed for a lot of their time period, we don't have much by way of an understanding of that period. So, yeah.
This is my first ever palynology question, so be kind. The Gippsland Basin, you were saying, has, is incredibly well understood, and I presume they've worked through all the assemblages of things, and it doesn't exist for Queensland to a large extent. Is that because the variation is this great through space or time, or is it because we just don't understand it?
So in the Gippsland Basin, what makes it so good for developing a biostratigraphic zonation is you had near continuous sedimentation through this time of types that collect and preserve palynology. In Queensland, we don't really have any one basin that we think captures the whole late Cretaceous through to this time period. So you can't just go, here are like 3 cores that capture the whole thing. We are going to have to probably go whole bunch of different things and line them up a bit like people do dendrochronology, where you go, this bit and this bit in these wells matches. So we're going to use this as that type of cross point. Whereas in the Gippsland Basin, because they have so much continuous sedimentation through a lot of this and really good seismic control, they can just go, here's a well, here's a well, and here's a well, and there is the Cretaceous to the Pleistocene, all in one big chunk. I mean, it is possible that some of our offshore tertiary basins could prove to contain much bigger sequences than we generally consider them to. Let me just see if I can get back to this one. Where is the map of the Cenozoic in Queensland? These are mostly the onshore. There's a couple of, is it the Rockhampton Basin? There's a couple of offshore basins that do appear to, according to the seismic, contain fairly thick sequences of rocks of this age, but I think a fair few of them haven't really had any drill holes put into them, and therefore aren't available to us currently for research. But yeah, but down south, finding the Gippsland and going, man, that's a lot of oil, really worked out well for them. Yes, Jen.
Yeah, so that kind of segues into the question of, there's a lot of oil, therefore a lot of biostrat in the Gippsland Basin, whereas a lot of these tertiary basins were indeed oil shale targets until there was a moratorium. But what other critical minerals might be lurking in associated with the hydrocarbons in these basins?
So one of the suggestions in some places has been a few of the rare earth minerals could potentially be preferentially sorted into some of these sorts of sediment deposits. Rare earth mineral exploration in Australia is a big emerging thing, but it's very much in its earliest stages. So at this point, we kind of don't know what we don't know. But yeah, these are some of the areas that are on the, not necessarily people working on them now going best targets, but they are sort of on that periphery of people starting to think about where you might find concentrations of some of the weirder rare earth minerals. Yes.
There was a question online.
Oh, sorry. Again, pardon me for betraying my ignorance here, but there's some work being done here in the SMI where it looks at how plants hyperaccumulate certain elements. Like we know there's work being done on cobalt, there's a bit of work done on gold. So I'm assuming that prehistoric plants, some of them at least, would have had similar capabilities. And so you would have a large presence of big forests that would be associated with deposits of some of these elements. Am I thinking in the right lines here?
Okay, so my grasp of what plants actually are, other than the things that make your biostratigraphic markers, are developing. But from my understanding of talking with people like Joan, potentially yes. Like
It's unknown because people haven't looked for it. So we tend to look a lot at the microfauna, right? So linomorphs and sparse organic matter associated.
I have no idea what you're talking about. I'm sorry.
Right? People look at black rock and rather than at the cold. So it's an unknown quantity.
Okay.
And while we're talking about that, would you be able to comment on how the flora changed significantly in Queensland? So we talked about.
Yeah, so from the few sites we do have from Queensland through this period. Lining them up is quite tricky, but it does seem, in the early part of sequence, so like that early Paleogene, you do have rainforest that is a lot more extensively covering the state than we currently see. There is some signs that you do start seeing a shift to that more modern sclerophyllous type bush vegetation in different parts, probably at different times. So here's where the age control thing gets to be a problem, because people often have to go, this is a rainforesty type assemblage, therefore I assume it is older, or this is a more dry community assemblage, so I assume it is younger. But we do have a core from the Atherton Tablelands that indicates sort of your old style rainforests were persisting till very late. Equally, some of the cores from down in southeast Queensland, you're probably seeing that change to dry conditions starting much earlier than in those places. So that is one of the bits we're working out how it changes, and in different parts of inland versus coastal, north versus south, we're being able to actually line these up beyond going, is this change contemporaneous across the whole thing, or can we trace how things dry out?
Any more questions in the audience? Oh, we did.
Hi. So thanks for the presentation. Out of complete ignorance. So basically what I understood is that somehow if you go, for example, to the Gippsland Basin and you get a core, you get pollen from different species, and that tells you roughly what time those species were around in that area.
Yeah.
So I'm obviously oversimplifying things here, but there's kind of like a reference.
There's a couple of big, because they've done so much study, they've got a few cores they've gone, all right, we're consistently seeing these species appearing and disappearing at this order, and we can link these occurrences to that geochronological timescale through marine fossils and some absolute age dating.
Okay, so yeah, that was kind of my question. So how do you build that reference? So if you were to go somewhere completely new, you could see pollen and you can see species, but then how do you know like what time that relates to if you don't have a reference? How do you build that reference?
Yeah, so you start building it. Ideally, you want a few good long cores that will cover a good chunk of time. And then you want to see what the order of appearances and disappearances in those are, and if you can see some consistency. So that gives you sort of your relative age dating.
Yeah.
To then link that back onto your geological timescale, where you start going number, you need some sort of tie point. Historically, a lot of that happened through links to marine biostratigraphic zones, which were tied then to the official boundary points of where we define the geological time periods. More last 50 years, we start using geochronological, so radiometric dating, where you have, say, an ash fall partway through in a few spots, that you can get an actual depositional age for that and go basically start pinning your relative age control method to the absolute time scale. And that means you can take that relative dating method and apply it to sediments where you don't get any convenient volcanic conclusions and go, we're pretty confident this is this age.
So I guess that's a work that needs to be done.
So it's why I think those Duaringa strat cores are potentially going to be a really good starting point, because at 1,000 meters of sediment, you're probably catching a good chunk. of this time period. And that should let you start tying some of these shorter cores into that and lining things up. So ideally, if you get really lucky, you find a basin where you have that really deep continuous record and you can just go, here are 3 cores through different parts of the main depot center. I have consistent appearances and disappearances through these horizons, and this is how I will put together the zones. Other times you need to use, more bits, but.
Oh, okay, no, thanks. And then very short question that the answer might be, no, it makes no sense. I'm from Chile, and sometimes I do see that the vegetation looks similar to the south of Chile, like araucarias. We have araucarias, and here I see a couple of trees that look like araucarias, monkey puzzles. So I'm like, is it worth like doing something over there, like or in different places where there was a like history, I guess, at some point?
So for this time period, you probably wouldn't be able to directly get the biozones used in Chile or those areas to apply here, because you'd run into broadly the same issues as you do with the Gippsland Basin. They're just that bit too far apart. But generally, when you're comparing any paleobotanical records of this period, to your near neighbours, Australia, New Zealand, Antarctica, and South America are sort of the sections where you first look for what is happening in your geological nearest neighbours, because you were starting with the same sort of vegetation groups going in, and they start splitting apart. But there is some evidence that there was interconnectivity to a greater or lesser extent till reasonably late in the process. So you couldn't necessarily borrow the biozones across, but some of those big trends and how they match up and how they differ are things that people get really into looking at.
So I remember this is a geochronology question, because you mentioned you had found a detrital zircon. I remember when I started working in much older terrains, the typical thing we would do is map a whole area in lurid detail and get like 3 age dates and sort of build this enormous house of cards associated with those very small amount of age dates, and then find out later that the house of cards was completely wrong, and that there were some rock sequences that were hundreds of million years different in age, and we just thought they were the same because they looked the same. My question is the same sort of revolution going on in terms of being able to place absolute dates on these basins? So, working with detrital zircons or other minerals that we were never able to date, but now we can date. Is that something that can help in this problem?
Yes. So, in particular for a lot of the Australian sequence, we have often had to rely for this period on dating the underlying integrated basalts and stuff. And our ability to get good geochronological dates out of those rocks in the last 20 and 30 years has increased massively. Like, there's a lot of older dates that are now considered fairly provisional, because our ability to get a good, reliable age has increased so massively. But like detrital zircons, in the event you can't get any syn depositional stuff, do at least help provide you a maximum possible age. Like, people are coming up with all sorts of really weird, wacky radiometric dating techniques that you can apply to increasingly sort of left field mineral groups that you can get included. I saw somewhere in WA, they've got a succession where you have sedimentary iron deposits from this period, and there are people who've worked out how to date those with one of the radiometric techniques. So like, there is a lot of potential. A lot of workers in Australia on the geochronological scale have really contributed to the development of those fields in terms of pioneering some of these techniques. And as a biostratigrapher, you know, your approach is generally to go, I will take whatever you guys think will work on my stuff. And increasingly, the geochronological toolbox is getting much wider. So a lot of them do only apply to fairly limited types of minerals and elements. But they're getting more and more tools that can apply to more and more different things. And it is really good because it is starting to let us put those, you know, hard tie points off. We have this biozone here, here, and here. And we've got, you know, actual age dates in different places. And you can see where in some, particularly in the Mesozoic biozones, you can see that some of them are not necessarily strictly the same age in different places. And you're really starting to get a much clearer way of tying it all together.
Very good. Thanks, Jen, for the presentation and initiating such a good discussion. Really appreciate you coming this morning. And so before we wrap up, just next week we have Doug Aitken, the General Manager of SMI ICE Chile. And we are lucky to be hosting him here in Brisbane for the next couple of weeks. And he will give us an update on the state of several projects across diverse topics that are happening in the center and their great impact in Latin America. Thank you.